The Center for Molecular Simulation brings together 7 different research groups with expertise in molecular simulation all under one roof.
The center was founded in 2012, but it builds on a long history in computational biophysics and computational chemistry at the University of Calgary. It brings together 7 groups in chemistry and biological sciences to study problems that require computer simulations from very detailed to much coarser levels.
We have research groups that focus on small molecules and electrons through to proteins and lipids, up to larger assemblies of lipid vesicles, all the way up to whole cells. We span the range from electrons to whole cells in the Center for Molecular Simulation.
We're able to treat many important biological and chemical problems using a very large range of time and length scales. Our main goal is to bring these groups together for research and training purposes.
My lab focuses on 2 different areas of protein structure research, and really what we're focused on is areas where we combine experimental data with computational models. And so on one hand, what we do is something called integrative structural biology, where we're trying to determine protein structures using sparse and ambiguous experimental data, with the ultimate goal of being able to determine the structures of those proteins, which will allow us to understand how they function at the molecular level. The other side of the lab is focused on protein design, and in particular on the design of protein-protein interactions.
We are working on methods and we're doing simulations to try to understand the physics behind properties of biological membranes. And the membranes give cells their structure. Membranes are basically the walls around cells, so they regulate traffic in and out of cells. If you want to deliver drugs to a cell, for instance in cancer, these drugs have to cross a membrane. We're interested in these physical processes, and we develop models to describe what happens when drug delivery particles move drugs to a cancer cell.
The main advantage of simulations is very high resolution. With the coarse grain model that we use, we can simulate fairly large membrane patches on long time scale because simulations become significantly faster. I use molecular dynamics simulations with the coarse grain models to understand the properties of lipid membranes at the most fundamental level.
I'm directly involved in making molecular models of nanodisk, which are alternative platform to study membrane protein. Basically, it's a disc-shaped membrane that is wrapped with scaffold proteins that allows you to have a membrane that is fully soluble in water.
Biophysics of membranes plays a key role in fundamental biological processes.
My laboratory is working on their selective ion transport across the cell membranes, and this is one of the main physiological processes and biophysical processes in the cell. Basically, we work on the understanding of how genetic mutations or effects of the drugs affect your heartbeat. So what we try to do, we try to understand on the atomic level first how the selective transport happening, how these proteins are capable of sorting ions, sodium, potassium, and calcium very, very rapidly.
We are using a multiscale approach. What is important about multiscale modeling is that we need to catch a balance point so that your modeling is computationally feasible, but at the same time, you don't lose information, relevant information of the nature of your system. This work is important, and this is illustrated by the fact that any malfunction of this channel caused by inherited mutation or drugs is associated with a cardiac arrhythmia.
We work a lot with physicians, as I mentioned, and pharmacologists in order to understand and to predict the potential cardiotoxicity of a novel drugs entering to the market before any expensive screening or human trials even, right? Just purely within silico models, because we can predict what are the consequences of the drug interacting with the cardiac ion channel will have on the heartbeat. Therefore, we can expand it for a very rapid screen, therefore decreasing potential for cardiotoxicity and very adverse side effects.
Having the center at the University of Calgary is fantastic. There's a very strong core group of people here, and it's a wonderful place to live and work. People are attracted because of the natural beauty of Calgary. It's a young, vibrant city.
I think the center is growing very fast. I think because it is so well defined, it's attracting new researchers.
I'm excited about research because we can push the boundaries of what we know.
You understand problems you previously did not understand. I think that's the most exciting way of measuring success. Understanding problems for which you had previously no understanding.